Hey everyone, it's time to start talking about insects again. If this is your first time here, or if you haven't already, please subscribe. It really helps me a lot, or like and share the video. Today I wanted to talk about some cool morphology things and how they link in with taxonomy. And this is an article that comes from the journal Arthropod Structure Development, which is a really neat journal if you've never seen it. And this paper that was recently published is about the fly Merodon. So in this case, it's Merodon mishustini, but it impacts the whole genus because the genus is a very significant pest of bulbs for flowering plants that you would normally plant in your garden, as well as things like onions and garlic. So this is a pest of international concern because it's mostly found in Europe, but we don't want it spreading internationally for the most part, although it has already broke quarantine many times. So this is the fly in question. It is a syrphid, and syrphids are usually referred to as like flower flies or hoverflies, sometimes bee flies, because a lot of them are mimics of bees. But it's not the adults that we're particularly concerned with, because the adults are actually fairly easy to identify. Now, generally speaking, how these flies get around the world is that they are accidentally shipped in the bulbs of plants like this. So this is a galanthus. But these flies can be found in lots of different hosts as far as bulb-forming flowers are concerned. In particular, we're also concerned with things like onions and garlic because they're so widespreadly grown, as well as a lot of international shipping is done with onions and garlic. So with bulb-forming flowers, the bulbs are usually what is shipped internationally because they are cold stored and people want to plant them in their gardens. Unfortunately, with Marodon, these maggots can burrow into the bulbs of the plants. So now you don't know that you have infested bulbs if you end up planting these in your garden. Now you have a Merodon infestation going. As you can see here, in this case, there's one large maggot found in each bulb. And these maggots, while they are syrphids, have a different look than most syrphids. Most syrphids look like this, and they are a predatory generally they're predatory species. They're kind of carrot shaped. So the head is here, it's more tapered, and the butt end is back here. The butt end usually has these giant fused spiracles, the posterior spiracle system that they use for breathing. They'll generally also have some anterior spiracles up here. But generally speaking, syrphids can be identified as being kind of carrot shaped, generally free living, very either wrinkly or spiky, and that they have these fused posterior spiracles. Their pupae are also very easy to identify. The puparium, again, has these giant fused spiracles on one end, and they're kind of teardrop shaped. Merodon is a little different, as you can see here. It's not obvious that this larva is tapering at all. And if you were to extract them from the bulb, you would find that there's very little tapering going on here. Generally speaking, Merodon is very either straight back and forth, or they're even kind of rounder, more stout, more bulbous. There's even a related genus called Microdon, which is even more bulbous, and it basically just looks like a dewdrop. You can still see that it has these large fused posterior spiracles that it uses for breathing. And up here it has this PPS is primordial pupal spiracle. So this is where the spiracles on the pupa will form, and up here are the anterior spiracles. And this is what passes for the head on these fly maggots. Generally speaking, with fly larvae, not all of them, but most of them have extremely reduced heads. And so all that's left there are some rudimentary antennae and some mouth hooks. So going off of these Merodon, they're easy or relatively easy to tell that you don't have a normal syrphid because they're generally inside something and you don't have this tapering sort of body. But the issue with Merodon is it's really, really difficult to identify them any further. And unfortunately, there are hundreds of species of Merodon. So right now we know of something like 234 species of Merodon worldwide. The majority of them are in Europe and kind of the Eurasia area. But unfortunately, we only have descriptions for 16 of the 234 species as far as the larvae and pupae are concerned. And the larvae and the pupae are what are normally intercepted in sort of the plant trade or the food trade. So this is a serious concern as far as identifying possible pests in material as they enter countries or as they leave countries, and this is a concern. And unfortunately, there just aren't that many people studying Merodon. So what they have done in this paper I'm talking about today is they've actually isolated this species, Merodon mishustini, and they have done a full exterior morphological workup in order to find characteristics that would be useful in helping to identify this species in its larval stage. And specifically, we're talking about the late instars. Generally speaking, with very young larvae, they're very hard to identify in most species because they don't have the full complement of characteristics yet. So these are going to be the larvae that are about to become pupae and then the pupae themselves. And what the researchers were hoping to do was find some key characteristics that will help identify this group. Now, spoilers, what they did find are very good characteristics for kind of the whole lineage. So this species and related species. Unfortunately, because flies don't have a great number of fly larvae generally don't have a great number of characteristics, it's hard to come up with singular characteristics that can be used to identify specific species. So first off, there are some really neat SEMs that they've collected. These are scanning electron micrographs of some of the characteristics of the face or the head of the fly maggots. So you have kind of the proto-antennae here. If you look at the mouth here, which is just made by these two hooks, you can see that the mouth hooks actually have these tiny little teeth on them, which not all flies have these teeth. But in general, the thing that we're looking at with this face is that it is a relatively short and round face or head. Generally, with other Merodon species, they're a bit longer, a bit more narrow. And in this case, it's kind of a bulkier head. They also looked at the anterior spiracles, which are here. So these would be the ones that are on top of the head up here. And the anterior spiracles poking out are quite something. You can see that the skin of these flies, the cuticle, are not smooth. They're extremely rough, covered in little spicules and granules and all sorts of things. So, and you can kind of see this with just a light microscope. You don't need to have a scanning electron micrograph. Or I'm sorry, you don't need to have a scanning electron microscope to be able to see this. Obviously, you won't see it in this sort of definition. But the key characteristic that they found with these Merodon anterior spiracles is that the openings to the spiracles here are not parallel. So in other related species, these spiracles are perfectly parallel, and in this group, they're oblique. So they're offset, and this can be used to narrow down to this group of Merodon. And you can also see there's all of these little hairs, these appendages that can be used for ketotaxy, but I'm not going to go into that today because it's a little much. On the posterior spiracles of both the larvae and the pupae, so this is a puparium here for this species, there are some interesting structures here. So generally with a lot of fly larvae and pupae, what you will see on the posterior spiracle are these big scars. So these circular things are spiracular scars, and these openings here are the spiracles. And on the syrphids, they are fused together, which in most flies they're separate, but they're fused together here on this large truncated cone. And below this ring here, you can see that it is covered in these little spicules and granules. These are a diagnostic characteristic for the group, it turns out. As far as we know, at least, like I said, only about 6% of the Merodon species have a workup like this. So it could be that other Merodon have this. But as far as this species and related species, this sort of structure down here, this base of the posterior spiracle, is covered with all this texture. And this is something that you can see with a light microscope and is apparently diagnostic for the group. But if we look at the pupa again, at the anterior spiracles of the pupa, what we see is, I think, much more interesting and much more definitive, which is these are the anterior spiracles, these cones, and you can see all the little holes that are used for breathing here. But in this group, you have this rounded anterior spiracle that is covered with these granules. For basically the first half to third are covered with these like warty granules. And the spiracle itself is curved It's mostly rounded, and this is an unusual characteristic for the group. So you might be able to use this characteristic of this warty, curved, kind of phallic spiracle in order to do a diagnosis if you are collecting these in the wild. So that's the utility of this sort of research into arthropod structure and development. You really do get to know the creature on a very fine detail level, and you can use this in other areas like agriculture or horticulture and things like that. If you all are interested in this sort of topic, I can pull more articles from this Arthropod Structure and Development journal, and we can talk about the kind of fine morphology. Just let me know in the comments, and I'll talk to you guys later.